Hi, my name is Mark and I'm a product specialist with Topogen. Today I'm going to tell you about some new cell reporters that we've developed to analyze epigenetic revisions, that is, changes in DNA methylation in a cell context. Changes in DNA methylation due to addition or removal of methyl C lead to changes in gene expression. Sometimes adding methyl C at a CpG is referred to as gene silencing. Removing it would be gene activation, just the reverse. Now, this revision in DNA methylation is not well understood, but can be assayed using some DNA reporters that we've developed. Just by way of background, we know that DNA methylation is a stable epigenetic encryption system. We know it's essential for life and development in animal cells. We know it changes as we age, and mistakes happen, so there are age-related changes in gene expression, which frequently are bad. And there are effectors of DNA methylation referred to as hypomethylating or hypermethylating agents. Sometimes they're drugs. They could also be other bioeffectors such as non-coding RNA. And we know that the drugs have been approved for treating cancer, and most would agree that there's an acute need for more DNA methylation effectors. So just to reiterate, we have new cell-based assays that have been validated for detecting DNA methylation revision that work in the context of a cell, and they can be used for identifying new DNA methylation targeting drugs, or the flip side of that is identifying druggable targets that can be used in treating chronic disease in humans. So let me tell you a little bit about how this process works. Now let me tell you a little bit about the basis for our reporter system. We found that if you have a DNA repair event inside the body of a cell, inside a context of a cell, using GFP as a reporter gene, and it undergoes non-homologous end joining or even homologous recombination repair, the cells convert to GFP positive phenotype. However, you find that there are an admixture of epialleles that are generated. Basically, some of the cells appear to be hypermethylated at the GFP locus, and other cells are hypomethylated at the GFP gene. And we can in fact sort these and generate individual populations of either heavily methylated GFP or unmethylated GFP containing cells. So for example, we would call these hypermethylated cells here in looking at this position as being DIM cells, because they're not very robust in their expression of GFP. On the other hand, we also have a population of cells that are unmethylated or hypomethylated, we'd call bright cells. Now, suppose you take a drug, say drug X, that induces hypomethylating activity, then you're going to get a shift in expression shown by this red arrow here from low expressing to high expressing. And this would be an example of an agent that would be like azadeoxycytidine, which inhibits the DNA methyltransferase. That would be an example of a hypomethylating drug. You can do the reverse experiment by taking the so-called hypomethylated high expressing cells or bright cells and test for drugs that induce the conversion to a lower expressing level, as shown by this green arrow. So this would be generating the reverse effect. So this defines a screen for our epigenetic effectors. And what we've done was we've isolated these individual cell populations, and they can be used as reporters back for other agents that induce revisions in imprinting or DNA methylation. Now I'd like to show you just a real quick live imaging 4 days after induction of a double-strand break, and you can really see what happens when the cells turn on and become GFP positive. And you'll notice that there are differences. Now here you see individual clones coming online through live imaging, and you can see that some express relatively high levels of GFP while others express low levels of GFP. And it's very clear from this slide that when you FACS sort these populations, you're going to be able to isolate a high expressor from a low expressor. A good example would be this cell right here is a high expressor. This cell here looks like a low expressor. So these can be fractionated and used as tools for drug discovery. This is another view at higher resolution of a single cell imaging analysis to follow these GFP positive cells. And what we did in this experiment was we took GFP and put a special GFP gene that was mutated. We put it into HeLa cells, and these cells are GFP negative because the gene is not expressed. It's been mutated, as I said. What we do is we subject these to DNA repair, and the cells become GFP positive. And the first cell that appears, as shown by this green arrow, is the parent cell or a progenitor cell. Now we know that this cell has successfully undergone DNA repair, and what we're actually looking at here is the analysis at a single cell level of the products of that event. So you can see here this GFP positive cell is going and it's producing two daughter cells, and one cell is going to be a high expressor or hypomethylated cell, and one cell is going to be a low expressor cell. And we can FACS sort these out and get individual populations from this primary event. This is another way of looking at the actual data on scatter plots by flow cytometry. And the easiest thing is just to look at B over here, where you see the GFP positive cells as a mixture of the low expressing cell population and the high expressing cell population shown here. Now you can do analysis with a combination of these, but like I said, we make it easier by isolating these individual cells by FACS sorts. So it's a little bit easier to analyze pure populations of cells, and I'll show you that in just a second. So here's an example of a hypomethylating drug called AZA-DC, azadeoxycytidine. And this is a population mixed of low expressing cells and high expressing cells. And what you can see here, if you look in this fraction, the green peak are the low expressing cells with no drug, and then the red shows low levels of AZA, and then the blue shows high levels of AZA. And you can see that these cells are shifting from this position as they're treated with the hypomethylating drug to remove methyl groups. They're shifted over to a higher position here in terms of expression. So that proves clearly that we're able to manipulate these expressing cells or these low or high expressors in precise ways using drugs of known mechanism of action. So here's how an actual screen would work if you had purified low expressing cells. Again, these are hypermethylated cells, and these are FACS sorted, so they're a reasonably pure population of cells. And you'll notice that we don't have any high expressing cells in this population, or very little, I should say. When you treat with a hypomethylating drug, again, in this case we treated with AZA-DC, you can see that a new peak appears, and this new peak is due to the conversion of low expressing cells or hypermethylated cells into a hypomethylated state, which gives you this readout. So if your lab is working on epigenetics and you have an interest in testing for DM effectors, this kit may be very useful for you for the following applications. You can do searches or screens for small molecules or synthetics, natural product extracts that inhibit or stimulate DNA methylation, and these would be classified as hypomethylators or hypermethylators with regard to mechanism of action. If you're interested in screening for pathway proteins or for bioeffectors, as a small non-coding RNA, microRNA, and for the actual pathway proteins, you would be looking for proteins or putting back genes that induce hypo or hypermethylation. This would be transfection-based knockdown or knock-in type experiments. But one of the things I want to tell you and emphasize is the single cell live imaging aspects of this tool, where you can actually add back factors and live image individual cells over time and get a really good idea how the drug or how the protein may be acting in the context of the cell. Now, so in summary, this is a cell-based screening system for DNA methylation revision. We offer this kit as the following components. We have detailed protocols on setup and validation. We have control drugs for reconstructing cell screens. We provide the cell lines, so they will be either low expressor HeLa cells for testing hypomethylating effector genes, RNAs or drugs, or high expressor cell reporter HeLa cells, and these would be for testing for agents that stimulate DNA methylation, in other words, convert the a hypomethylated reporter to a hypermethylated state. To order any of our products or to inquire further, please go to our website or contact us at support@topogen.com. We offer strong technical support for all of our products. International customers can see our distributor list on the website. And I'd like to thank you for your time.